In today's lesson, we're going to just quick review classification. Much of this that you're going to see is a review from Baby Bio. Okay, so there's very little here that's actually brand new. So first thing then for review, remember that the 7-step hierarchical classification scheme goes kingdom, phylum, class, order, family, genus, species. Remember that at the beginning of this, we also had domain, the D for domain. Yes, you have to still have this memorized. So if you can remember the mnemonic device, Dumb King Philip came over from Germany swimming. Dumb kings play cards on flat green stools. Any of those, that just helps you to remember in order what those steps are. Remember that this is hierarchical, right? So the largest category is at the top with the domain, right? And then it just goes down from there in terms of you get smaller and smaller categories. Now, while the categories get smaller, remember the degree of specificity actually gets greater as you move down that hierarchy from domain towards species. So as you get fewer and fewer organisms in each group, they have more and more in common within each group. So the things in the same genus are going to have more in common with than the things in the same family or the things in the same order. Okay. Just some quick reminders about that. Also, sometimes you might hear a question like, what's a group of related classes called? Well, a group of related classes is a phylum, right? Phylum is a group of related classes. A phylum contains all the classes underneath it. Remember, the phylum is the big group that contains everything underneath it. I'm trying to think if there's anything else very specific there that you need to know, and I think that's it. Some of the plurals of these terms are a little weird. So if you see the word phyla, that's plural of phylum, and genera is plural of genus. So it's not genuses, it's genera. If that pops up in an AP question, just so you know. Okay. Now, in terms of hierarchical classification, this is the scheme that kind of evolved over, several hundred years. It started with Linnaeus. During Linnaeus's time, classification was very hierarchical, starting with big groups that got more specific as you go down. But there were fewer steps in the classification scheme when Linnaeus was doing his work. And then over time, we've just kind of added more and more groups to the point that now we have, in fact, domain is the last addition to this classification scheme. And domain is like within the last 30 years or so where it was added in. So it's kind of evolved over time. But some scientists have a problem with this hierarchical scheme. And they want to do something a little different, something more descriptive and less hierarchical. And so it's been proposed that we use a phylocode, which would include more information about the organism's evolutionary history, its ancestry. But this is still controversial. This is kind of an ingrained scientific thing right now at this point, and I just don't see it changing anytime soon. All right, so domain, that's right here, right? There are 3 domains. That's the one that I said was added within the last 30 years. The bacteria are split into 2 domains. All prokaryotes are split into 2 domains. Archaea contains only kingdom Archaebacteria, and bacteria contains only kingdom Eubacteria. Okay, and then the eukarya is all the eukaryotic kingdoms. So that gives you kind of the basics of hierarchical classification. Don't forget also that you learned binomial nomenclature for scientific names in Baby Bio, right? Don't forget about that. If you are writing a correctly written scientific name, you can see here at the bottom how those are correctly written, right? You always capitalize the genus and everything else is lowercase. And if you're typing, you would italicize the whole thing as you see right here on this slide. If you were handwriting a scientific name, you would underline it. So you would see something like this, like E. coli. And by the way, with that E. coli that you're seeing there, Notice you can actually abbreviate the scientific name, right? It got kind of cut off there on the slide because my picture is here, but E. coli stands for Escherichia coli, right? But when you're working on a scientific paper or something like that, and you're writing Escherichia coli all the time, Escherichia coli, Escherichia coli, Escherichia coli, after a while it gets crazy. So you don't need to spell that out every single time. Instead, scientists just abbreviate it by abbreviating it with the first letter of the genus, keeping its capitalization, and then the specific epithet, which is the species part of this. One thing to remember about scientific names is that often this specific epithet is a description of a trait, or that's where you might see something named after a scientist who discovered it. So there are a lot of things that have the specific epithet Jeffersonianum, right? Because remember, Thomas Jefferson did a whole bunch of classifications. So either scientists who find things in and around Monticello name things after him, or things that are in Virginia in general got named Jeffersonianum after Thomas Jefferson. That specific epithet, because of its nature, treat it like more like a person's first name. So it's almost like the scientific name is written the way that you would write your name, like on an application, right? So mine would be Holly Audra, right? And while Audra is not a common name, if my name were something like Jennifer, right, you would have a lot of Jennifers around, right? And so this coli is kind of the equivalent of a Jennifer, or a Nick, or a Sam, or a Jack, right? Those are common male names that you find. Just because two individuals have the first name Jennifer or Jack does not mean that they're related. They would be more likely to be related if they also had the same last name. So this is kind of written with last name first, then the first name is like a specific epithet. Okay, so don't forget those things about scientific names. All right, there's two schools of thought when you are doing classification. There's phylogeny and then there's cladistics. Okay, so phylogeny is kind of the preferred method. This is studying the entire evolutionary history of an organism. And you're tracing that with phylogenetic trees. And phylogenetic trees are what you did in your POGIL the other day, where you're looking at all the known data for classification and you're branching according to the hierarchical classification schemes. Some drawbacks are that the branch points aren't always accurate with respect to time, but you can actually get a sequence of events from a phylogenetic tree. So you may not know exactly the time that a particular trait evolved or particular species evolved, but you can see relative to other species when something evolved. It is also kind of hard sometimes to tell evolutionary relationships between certain taxa on phylogenetic trees. Sometimes they can get really busy too. They can get really complicated to look at them. So that can be a drawback. And you can't necessarily infer that an organism directly evolved from the taxon next to it on a tree. In general, when we have branch points on a tree, we call those nodes, and the node is a theoretical common ancestor that may or may not exist. Okay, we just may not have found it yet, or something like that. So there are some drawbacks. Cladistics is a different approach. It's not terribly different, but it's slightly different enough. You're using common ancestry as the primary criteria for classification, but then you construct the cladogram using cladistic analysis, using derived characters. Okay, so you're using the appearance of specific characteristics that were derived from some common ancestor to use your branching. And when we do our BLAST lab, you looked at a cladogram and looked at how those are constructed. Okay, so derived characters are what we use. In cladistics, when you're doing a cladogram, you're only using a bifurcation, so you don't have splits like a phylogenetic tree where you could have 3 or 4 things evolving at the same time from a common ancestor. You really split cladograms one of 2 ways. So there are some drawbacks, but they tend to be a little cleaner and neater to look at. And sometimes when you're just focusing in on one group of organisms, it's useful to look at a cladogram instead of a phylogenetic tree. Anyway, so there's some important information about cladograms and phylogenetic trees. All right, so there are also a couple terms that are important. Most of the taxa in the classification scheme, there's another funny plural. Taxon is a single step in the classification scheme. Taxa are multiple steps. So a kingdom is a taxon. Kingdom, phylum, class, order are all taxa, right? And those taxa are meant to be monophyletic, meaning they have a common ancestor, right? That's the goal overall of classification, is you're grouping things based on their evolutionary history, based on their phylogeny. So you're trying to get to these monophyletic groups where you have a common ancestor. There's a node, there's the common ancestor. This is a monophyletic group, common ancestor there. Altogether, this would be a polyphyletic group because if you're looking at the whole cladogram, it's polyphyletic because you've got several common ancestors there. Okay, so this group is more distantly related to that group. All right, so when you are doing classification, the greatest thing that we're always trying to accomplish is distinguish between homologous features and analogous features. Okay, homologous traits, are traits that are similar due to a shared ancestry, where analogous traits are similar due to convergent evolution. In other words, a trait happens to just be advantageous in a particular environment, right? So it pops up and you see it in multiple groups of organisms. So wings are particularly useful, which is why you see them popping up in insects, you see them popping up in birds, you see them popping up in bats. That does not mean, however, that they came from a shared common ancestry. They're analogous traits, right? Homologous traits would be they would have come from a same common ancestor. And this word homoplasy, I don't know that that's a College Board term that you have to know, but that's a term for an analogous trait, is a homoplasy. But in general, these days, it's really hard to distinguish when you're just going with physical characteristics. It's hard to distinguish whether that trait is due to a common ancestor or if it's just an analogous trait. And so for the most part, what we do is use molecular data now to do classification. It's a lot more accurate, just because we know about genetic codes, and we know about the normal mutation rate, and we can actually trace through time just based on mutations, differences in the genetic code for certain things. We can trace that ancestry backward if we need to. And you could also use protein sequences, right? So you can either go to the source code, the DNA, or even RNA sequences if you want, or you can go to the product of the source code, which is proteins. So either of those works. So if you're looking at the amino acid sequence of a protein, that also works because mutations over time would cause changes in particular proteins. Okay, so we can look at molecular data to help us. But then that kind of helps us to inform what we're seeing in the anatomical or physiological data. All right, so here's a great example of a homeoplasy. Here's a common dolphin, and then here's an ichthyosaur. Notice these are not monophyletic. Look, here's the ichthyosaur here next to lizards, right? And we know that lizards, the reptiles, have some common ancestor with birds, right? And things like that. So this obviously is in no way related to mammals, right? Which here are the mammals, right? Whoops, sorry. No, we need to go back to the mammals back here. So there's just no relationship between them, no close relationship, no close common ancestry there. Okay, so that's analogous. Even though they look very similar to one another, that's due to convergent evolution because they both live in aquatic environments. So certain physical traits are just advantageous in that environment, and that's why they look alike. It doesn't have to do with any common ancestry. At the bottom, you can see where there's common ancestry because you can compare these particular genes, the human and the fruit fly genes, to see that there is some common ancestry there. All right, now here's the thing. Classification is just darned hard, right? For years and years and years, all we had to go on were physical traits. And then with the advent of the discovery of the structure of DNA and our ability as time went on to sequence that DNA molecule and be able to read the code and then take that and read it into proteins and so on, that has greatly modified how we've looked at classification over time and how we've looked at the classification of specific organisms over time. So we went from looking at physical characteristics, which we've already said are really deceptive, to now modifying a whole lot of classification just because of the molecular data that we've got. But there's still a certain arbitrary nature to classification. When you were looking at your blast lab, I hope you saw some of this. Even when you have molecular data, if you're looking at that percent coverage and that percent identical, sometimes there are several species that are identical, and you had to do a Sophie's choice to try to figure out, okay, which one is the one that I'm going to choose for my lab, right? So there's a certain arbitrary nature to this sometimes. And so the key with classification is to always remember that when you are looking at a diagram, any phylogenetic tree, any cladogram, it's somebody's hypothesis. Okay, it's like a schematic of their hypothesis. It's a visual of someone's hypothesis. Don't look at that as theoretical. Don't look at it as set in stone law. Okay, it changes, and it changes all the time. Okay, even though it doesn't make the news, it's not a sexy topic in the news when something changes classification. It still happens. Okay. Scientists will use several things to try to be as accurate as possible sometimes when they're drawing their phylogenetic trees. That's called achieving maximum parsimony. Some phylogenetic trees will use proportional branch lengths to demonstrate the degree of change since the two species evolved from a common ancestor. But then there's also good old Occam's razor. I don't know if you've ever heard of this. It exists in statistics and it exists in science. When you have two competing hypotheses to explain a phenomenon, you would select the one, if they both appear to be equally plausible, you select the one that is the simplest with the least amount of assumptions involved in it. Okay. Now, does that mean it's the right one? No, it doesn't. It just means it's the one you're going to accept and use. Okay. And then some scientists use the principle of maximum likelihood, given the rules of how DNA changes over time, constructing a tree based on that. Again, I mentioned we know about normal mutation rates. We can construct evolutionary clocks based on those mutation rates. So when we see the number of mutations, we know about how long it's been since two species diverged just based on the number of mutations. So you can use that information as well.